5.1 is for the saturated salt solution. And so these halophiles have pigments which protect them from the sun. And also some pigments which were called rhodopsin, which will help them to capture the sunlight for energy. So halophiles, many halophiles can phototrophic. They can get their energy from the sun. So this is very important. Okay, the least salt concentration 1.5. The highest is 5.5 molar. Okay, so hypersaline environment are abundant on the Earth's surface. One big one is the Great Salt Lake, which is in the American Middle West, called the Utah State. This is the Great Salt Lake, which originally was a sea and then evaporated to almost the saturated sodium chloride concentration. The green color here is the algae, halophilic algae called the Duna liana. And here we can see the concentration of ions in the Great Salt Lake. It's similar to its relative composition is similar to the sea. But in here, the lake Zugum, we can see the pH is very high. So this high pH is because of its bedrock is made of calcium carbonate. So carbonate dissolves into it and grab hydrogen ion into it, and so it became leaves hydroxide ion. So hydroxide ion elevated the pH to 11. So this is why we can have both very alkaline and both very salty lake in the natural environment. And this is in Egypt. This Lake Zugum in Egypt. It's called Wadi El Natrum. Natrum is soda. So this is called a soda lake. So in soda lakes, we have both high salt and a high pH. Soda lake, okay. Duna liana, extremely halophilic. Okay, so this environment leaves many halophilic bacteria. So scientists are interested in how they can maintain the inside and outside osmotic pressure. So these are this evaporation pond that is for making the sea salt for human consumption. It's a natrum lake. The red color is from the Rhodosphaerum. Rhodosphaerum is red-colored bacteria. So these are some of the halophiles. Their cell shape is like a brick. Okay, so they need the sodium for growth. And their counter ion, that is how they deal with the high salt is that they maybe accumulate potassium ion in their cell body up to about 4.5 molarity more. And sometimes they accumulate compatible solute. Compatible solute means the solute is able to stay in the cytoplasm, but will not harm or hinder the metabolism. Some compatible solute are glycerol or betaine this kind of material is organic chemicals, and they can accumulate to high concentration in the cell, or some use the potassium ion as a counter ion to counter the sodium on the outside. So their metabolism is quite interesting. So when sodium is not present, the cell will lyse. The cell will lyse. Okay, and so these are the very important physiological characteristics of the halophiles. Okay, so they are generally mesophile. That is, they don't like high temperatures, and some live in alkaline conditions. So these are the general characteristics. That they have the bacterial rhodopsin or halorhodopsin. So these are the bacterial protein pumps on the membrane, which can increase the protomotive force. And also other kind of rhodopsins can direct their photosynthesis or direct the flagellum. Okay, so these are the color. These endow them with the color for the archaea. Okay, let me show you the picture. This is their bacterial rhodopsin. And with the sunlight, they will get the hydrogen ion out and to produce a protomotive force. So this protomotive force in here, when they flow in, then will generate ATP using the ATPase. Also, they can also promote the turning of the flagella so that they can swim in their environment. So they are red colored or pink colored. Sometimes if you place a salted fish in the refrigerator for a long time, then you can see some red spot, huge red spot growing on the fish. And this red spot is the halophile. That is contaminated from the salt, from the sea salt, which are used to salt the fish, preserve the fish. Okay, so the halophile is the one major category of Euryarchaeota. Another big category is called methanogens. That is, they produce methane. The reason is that they use organic material for energy, but they dump the hydrogen, dump electrons into carbon dioxide or other methyl groups, and so reduce this carbon dioxide or reduce the methanol to methane. So methane is as a reduced carbon was released as waste. So biogenic methane was discovered by this Italian physicist called Alessandro Volta. So this is the modern collection of the marsh gas. Marsh is a place where we have lots of anaerobic sediment. If we put a bottle, water-filled bottle into the mud, then the gas produced will be gathered, will be collected in the bottle. Then if we retrieve the bottle and light it, then the gas will burn. So this is the marsh gas. So marsh gas is used in the countryside for cooking or for generating hot water or even for electricity. So this marsh gas energy source is useful in the rural area, in the countryside. And so Methane can also generated in the rice paddies, also the landfills, the wetland in this place can also generate a lot of methane. In Siberia, tundra began to melt. The tundra is a frozen soil, and because the climate, global warming, those tundra gradually was thawed. Then the methane, the frozen methane was gradually became gas, and those gas sometimes appear suddenly as a huge hole on the plane. And this huge hole is a huge volume of methane getting into the atmosphere. So this kind of big huge methane hole was observed in Siberia for the past tens of years. Besides these places in the digestive tract of termites, cattle are also the source of methane, even in human beings. So these are the various sources of methane. So the methanogen can use carbon dioxide or acetic acid or methanol or methylamine, about 11 kinds of organic material, and then convert them to methane. You see here methane, here methane, methane, methane. So these are the major 11 substrates which can be used by various kinds of methanogens to be converted into methane. The methane was either used by methanotrophic bacteria, or be released into the atmosphere. Okay, so some bacteria can use the waste released by another bacteria. So this kind of relationship is called syntrophy. Syntrophy. That is, they eat together. It's called syntrophy. Okay, habitats of methane is landfills, swamps, marshes, and in the animal digestive tract, and geothermal sources of hydrogen carbon dioxide. So these places are the place for the methanogens to live. They are absolutely anaerobic. That is, only the very oxygen-free places can they live. And in the countryside, people will gather the manure, gather the feces of pigs, cows, animals, even human beings, and then put in an enclosed tank or enclosed structure without the air. And then in that place, methanogens will grow and release the marsh gas. And if farmers collect those marsh gases, and then can be used for cooking as a gas, as burning gas, as electricity, for generating electricity. Okay, so this is methanogens. Besides the methanogens and the halophiles, there are some kinds of the thermophilic eukaryote. So such as the Thermococcus, Pyrococcus, is kind of archaea. That is, although they belong to Euryarchaeota, but they love heat. They are thermophiles. Okay, so these are the thermophilic archaea. Okay, these are the habitat for the thermoplasma. Thermoplasma is a thermophilic archaea. This is called core pile. That is the residual core which was not used after they are mined. So they are put into huge piles. If water seeps in, then the thermoplasma can grow in it, which will generate the heat up to about 50 or 60 degrees. Okay, so these are the thermoplasma. And some of those habitat for these thermophiles are these places, right? These places are collectively called sulfatara. Places where the sulfur is part of the environment. It's called sulfatara. Okay, let me show you more of kind of sulfataras. More of those kind of sulfataras. Oh, this one. This is the sulfur pool. That is the underground gas comes out here, which contains hydrogen sulfide. Hydrogen sulfide was oxidized to sulfur, elemental sulfur. So this is hot water mixed with elemental sulfur. This is a sulfur pool. Okay. And another kind of Euryarchaeota is called the barophiles. Barophiles is also a kind of eutrymophile, but they can withstand the high water pressure. It's called the barrel. And the general bacterial, especially marine bacteria, is able to withstand water pressure up to 400 atm, atmospheric pressure. We know that every 10 meters of water will increase 1 atm pressure. That is, as we dive down the ocean, then the water pressure, hydrostatic pressure increases very rapidly. That is 1 atm per 10 meters of seawater. Okay, so general bacterial, terrestrial land bacteria can tolerate higher pressure. It's called a barotolerant. But there are some extreme barophiles. Extreme barophile means that they can withstand water pressure up to 800 to 1,000. 1,000 means 10,000 meters underneath the water. So these are called extreme barophiles. Once they are out of this high pressure, they will burst. However, for those barophilic bacteria or archaea, they like the higher pressure. But they can still survive in 1 atm. 1 atm is the pressure on the surface of the land, on the surface of the water. And some archaea. See the percentage archaea. The percentage archaea increases with the the water depth. That means that archaea on the surface bacteria is more than archaea in terms of number, but in very high depth, bacteria and archaea are about 50%. That means the diversity of archaea increases with increasing water pressure. So these are the exploration of the deep sea. That is, this is the mechanical arm to dig out the soil, dig out the sediment. It's the environment in the hydrothermal vent. And these are the tube worms living around the hydrothermal vent. So they have a lot of bacteria living together with them. So these bacteria is able to capture the sulfur for the energy source. So these are these have the organisms for the sulfur. Okay, the one most common archaea in the hot spring is called sulfur lobalis. That is, it's a low wood bacteria. That is, it's one bacteria which is soft, coccus-like bacteria. It's called sulfur lobalis. This is quite often recovered from hot spring, sulfur hot spring on the surface. So I have also identified one called the Sulfolobus yangminensis, that is in honor of the Yangming University. And that is from the hot spring in the Yangmingshan region. Sulfolobus is like this. So their virus is like this. Okay, so these are the terrestrial Craniobiota, and others are these are the archaea which are in the Euryarchaeota, called ferroplasma or Picrophilus. So these are the like a thermoplasma, but they like very low pHs. Okay. People have collected samples from the ocean bottom, especially near the black smokers. And so they have recovered, or they have cultured some bacteria. It's called the vent-adapted Craniobiota. So these are the very interesting ones. First, they are extremely anaerobic and extremely thermophile. So the typical ones are called pyrodictyum. Pyro means fire, right? Dictyum means web. So this is called a fire web. And after they are cultured in a very high temperature and anaerobic environment, then we can see that they form a disc and interconnected by the small fibers. It's called a cannulae. Cannulae means a miniature canal. It's called a cannulae. So these they form a network of archaea. So the name is called a pyrodictyum, fire net. And then looking under the scanning infrared microscope is like this. So if they can, when they divide, they produce many cannula, and this cannula interconnects with each other to form a network. So this is very interesting archaea, which of course is very difficult to culture. Okay, then let me show you another very interesting archaea is called a nanoarchaea, which is very small. Is also a parasite. It's a parasite. That is, it's a symbiont. It's a symbiont of Echincoccus. Echincoccus is another kind of archaea. It's Echincoccus hospitalis, and it grows on it. Let me show you the picture. This picture appears in Nature. That is, it is so interesting that Nature makes it cover, makes this picture a cover on the Nature. That is, Echincoccus is in the center. Then this nanoarchaeum is symbiotic with it. So people think that it get some nutrient from the Nyctococcus. So they may be the parasite in the archaea. Okay, the near the final ones called the Methanopyrus. That is, it is a very thermophilic bacteria, but it can generate methane. It generates methane. So people think that this is at the very origin of the phylogeny. That is, it is also not only produces methane, but it's very similar to Crenarchaeota. And also recovered from hydrothermal vent chimney that is 2,000 meters underneath the sea surface. So this Methanopyrus can be cultured, but must use a chamber which is almost 90 degrees Celsius and without any oxygen. Okay, so finally, let me talk a little bit about the Thalma-Archaeota. Thalma-Archaeota is ammonia oxidizing archaea. Ammonia oxidizing archaea. Okay, so this is very interesting. Okay, besides those typical ones, some Crenarchaeota also is found in moderate habitats. Moderate habitats, that is in the sea surfaces. Sea surface also can be also in the soil of plant. So in the past 20 years, scientists are able to find more and more archaea in milder places. So besides those extremely environment, archaea also exist in milder places, such as the area around Antarctica, or in the deep sea, or in the surface of the sea, and also on the soil, these kind of places. But currently, there's not any pathogenic archaea being discovered. So we have to be comfortable that when we deal with them, we don't need to worry about their pathogenic effect on the human beings. Okay, let me see if I have any more virus. Okay, finally, let me mention some biochemistry about it. That is, there are proteins that to be very stable or thermophilic. And the reason for their thermostable proteins are the hydrophobic core. The core of the proteins has to be hydrophobic, but the surface has to be ionic. And there are some chaperones or thermosomes which help this protein from being denatured. And this DNA is stable at 110. The reason may be that it has this one, diphosphoglycerate, to that is to kill it with this DNA or the DNA binding proteins to protect the DNA from heat denaturation, because some of them can withstand heat as high as 120, 121, and also have some archaeo histone. Histone is a DNA binding proteins. So with the discovery of these proteins, so scientists are trying to find a reason why the DNA can function at 120 degrees Celsius. And finally, its energy. Its energy may be from the hydrogen. we form the hydrogen. So the archaea is a new branch of microbiology, which attracts a lot of scientists to study them. They are interesting in itself, and also they are interesting for our understanding of the microbiome evolution. Currently, people think that hyperthermophiles are actually the original species of microbes on Earth, because they may be generated from the hydrothermal vent in very hot environment under the sea. Okay, so I have very briefly introduced microbial ecology as well as archaeal diversity to you in this lecture. And some are very brief. And if you want to have more information, please read the PPT as well as the textbook. So do you have any question now?
No.
No question. No question. As for the test, I will not ask questions about the individual bacteria or individual archaea. Only the general properties, or only the general knowledge will be tested. So you don't need to worry about all those names or the properties of each individual bacteria or archaea. So don't worry about it. Just reserve some time to read, and then you will profit a lot from microbiology.
Okay.
So tomorrow we'll have Dr. Huang giving the 4th lectures about the bacterial physiology, about the growth, and the other aspects of the microbiology. And the microbiology has undergone a lot of changes in its research activity. In the 19th century, it is finding the pathogens, identifying the pathogenic bacteria. And in the early 20th century, it is finding the environment, microorganisms. That is the general microbiology. Another trend is the microbial genetics, which gave birth to the molecular biology. And at the end of 20th century, it is the DNA, the study of DNA, study of DNA machinery, as well as the DNA sequencing. And the first 20 years of the 21st century, it is the metagenomics, or the genomics, or proteomics, that is studying the totality of the microflora. So you can see that with the changing in the direction of research, so we teachers have to modify our course material so that you are able to read scientific journals. They're able to understand it. And so this is why we can only teach a part of the whole field of microbiology to you. But if you are familiar with the basic of the microbiology, you can go back to the old literature to understand their activity, and you can also read the newest literature to know where the research direction is. So have you heard about the CRISPR? CRISPR is one new direction in the study of microbes. That is, the microbes have immune system in their small cell body. This immune system uses only a few genes to protect themselves from invasion by virus or plasma. The way to do it is that they cut off the invading DNA and try to copy a section of the invading DNA and then paste it in small section between the interspersed sequences and the repeating sequences. So this is a sign that says that I have known you, and I have a piece of your sample in my body, in my genome. So their defense is that they will make a replica of this piece of foreign DNA. So and move this foreign DNA to this RNA, transcribed RNA, to couple to find the invading DNA, and then use the endonuclease in this machinery to cut up the invading DNA or invading RNA. So there's such a miniature immune system in the cell body, in the bacterial cytoplasm. So this is a very tremendous discovery, and generated a Nobel Prize for two female scientists. One is called Charpentier, another is called Doudna. So you can try to find their description of them on the YouTube or in the Google. So their discovery is very exciting because we can try to modify the DNA, modify animal DNA by using this machinery. It's called the Cas9 machinery. So this is a new direction. And this new direction in the microbiology actually helps the whole biology with a tool to change the DNA repertoire of animals or plants. So this discovery is very exciting, and also totally grown out of microbiological investigations. Okay, so so this there are many more interesting things in microbiology. However, because of the selection of the topic, we have to ignore it, and hope you have time to get literatures on these new discoveries. Okay, this is about nearly 11:19. That's a little bit past our time. Okay, so I hope that you can read the PPT, and if you have more time, please try the textbook to read the textbook and to enjoy your exploration of the microbiological world. Thank you very much. Thank you very much, and hope that I can see you later on in Taiwan or in Vietnam. Thank you very much. Bye-bye. Bye-bye.
Thank you, sir. See you somewhere on the shore.
See you in Taiwan or in Vietnam.
Yes, sir.